Hello everybody, this is going to be my first of 3 videos on photosynthesis. This is the stuff that we covered either of Monday or Tuesday of this week, and then I'm going to go into the light reactions and then a separate video for the Calvin cycle so that they're a little bit shorter and a little bit more digestible. But let's go ahead and start with these. So we started talking about photosynthesis by discussing redox reactions. So when we say redox reactions, we mean reduction-oxidation reactions. That's what this first part stands for, the RED for reduction, and then OX for oxidation. Now the trickiest part of this is remembering which is which. And so an oxidation is a loss of electrons, whereas a reduction is a gain of electrons. And I know that seems sort of counterintuitive to what you think, right? If you were to reduce, why are we gaining? Well, it's because it's dealing with the charge. So remember that electrons are negatively charged, so if you gain electrons, you are reducing your overall charge. And so one of the ways to remember this is with oil rig. And so oxidation is losing, stands for oil, and then reduction is gaining, stands for rig. And so it's up to you on how you want to remember it. For me, I personally just remember that it's going down in charge, so it has to be gaining electrons. But this language ends up being very important as we move through photosynthesis, as we talked about this week when you saw NADPH. And it'll be very important when we talk about cellular respiration as well. So once again, you're never going to have to balance any sort of equation with electrons as you might do in AP chemistry, but you do have to understand what the verbs mean. When I say that this molecule is oxidized, you need to understand that that one is losing electrons, or this molecule is reduced, this molecule is gaining electrons. And we saw that was very important when we talked about the Calvin cycle this week. Okay, so now let's go ahead and start talking about photosynthesis specifically. And so there are some review terms that you probably remember from last year. Autotrophs, remember auto meaning self. These are the ones that are doing photosynthesis. So we consider plants autotrophic. We consider animals heterotrophic. Now there's others in these categories. For example, algae. Algae is not a plant. I know we often think of it as a plant. It is not. It is a protist and oftentimes single cellular, whereas plants are typically multicellular. Bacteria can also be autotrophic, specifically cyanobacteria. And then heterotrophs, it's not just animals, fungi are decomposers, so those are considered heterotrophic, and a lot of bacteria also are heterotrophic. These are just terms that you need to be comfortable with. One, because you'll see them on the AP exam and they will just assume that you know what they mean. And two, I will do the same thing. I will just say, oh, autotroph, and I expect you to know what I mean. Okay. And so this is the chemical formula for photosynthesis. Now, I would love to stop here and just say that's all we need for this year. Actually, I wouldn't, because then you'd be missing out on some really cool stuff. But as you can imagine, it's a whole lot more complex than just the overall formula for photosynthesis. But it is important to understand the inputs and outputs overall, and then the inputs and outputs for each individual reaction that we talk about. You also need to know where these are sourced. So the way I like to think of photosynthesis and cellular respiration is to think of it like a recipe. And so I need to know what I need to know how to get it, and then I need to know the steps to get there and the machinery that I need. All of that stuff will tell me how I get from A to B. So for photosynthesis, I need water, and that's going to be sourced from the roots. So plants will have roots, and they'll bring water into the tissues, into the cells, and ultimately to the chloroplast. I need carbon dioxide, and the carbon dioxide comes from the atmosphere. Then we also need light, and the light is coming from the sun. Now I also want you to remember that if I say photon is a light particle, and so you need to understand that I'm talking about light in that instance. And then what do I get out of it? Well, I get glucose, which is a sugar, and that's going to be used for cellular respiration. This is why photosynthesis evolved. It evolved so that it could store energy in the form of a carbohydrate. Oxygen is just a byproduct. It's not the reason that it evolved, but it's a byproduct that we love and lots of other organisms love as well. So how do we actually get the CO2? Well, we said it was from the atmosphere, but specifically, how does it get into the cells? Because plants also have dermal tissue just like we do, and so that is going to prevent just anything from getting into the plant. And so they need to have specific openings that allow gases in and out. And these openings are called stomata. So stomata are going to be these openings that will open and close because of these guard cells. And when they open, CO2 is able to enter. Unfortunately, that also means that water and oxygen leave. Now, oxygen leaving isn't a huge deal. Now, some of the oxygen is going to need to be used for cellular respiration, but generally we want to diffuse some of that oxygen out. But water, we don't want to lose that. Water is very, very important, and so they really, really want to conserve water. And so this is going to be a balance. The plant is going to have to open and close according to both CO2 needs and water needs. That becomes very important, and we talked in class about a lot of the adaptations that plants have evolved in order to prevent water loss, such as a waxy cuticle, deeper roots, more widespread roots. Those are all very important. It's not so important for this specific unit, but just in general, the anatomy of a plant can be really helpful for understanding both this unit and when we get to evolution. Okay, and so now let's talk about the two main stages of photosynthesis. We have the light reaction and the dark reaction. Now, I told you I don't really like the term dark reaction. It's getting outdated anyway, but if you see it, I want you to understand that that is the Calvin cycle or the light independent reaction. The light reaction is the photo part of photosynthesis. So it is going to take a photon and do some work with it to ultimately get ATP and NADPH out. Then that NADPH and that ATP is going to be used in the Calvin cycle to synthesize glucose from CO2. So that's why we say the Calvin cycle or the light independent reactions are the synthesis part of photosynthesis. So I used this as a bell work the other day, and I think it's important to understand that photosynthesis is only going to increase to a point of saturation. Light, carbon dioxide, and water, we can't just keep adding in more and more and more and assume that photosynthesis will happen forever. And if we increase the temperature, we actually have an optimal temperature. Once it's super, super cold, photosynthesis doesn't really happen. When it's this sort of optimal temperature, photosynthesis is happening very quickly. But if we go too hot, then photosynthesis actually decreases. And we've actually seen these types of graphs before. We saw them in the enzyme section, which means that it is very likely that photosynthesis is highly regulated by enzymes. And we're going to see that later on when we talk about the light-independent reactions and the light-dependent reactions. But I do want to plant that in your head now, that this is highly regulated by enzymes. And so those enzymes are going to only work so fast. It doesn't matter how much carbon dioxide or how much light you give it. To a certain point, it will only go so quickly. Okay. So now I want to know where these reactions take place. They take place in the chloroplast of the plant cells. You should know this. You probably learned this last year, but it's always good to remind you that it happens in the chloroplast. And these chloroplasts are organelles that contain chlorophyll. That's why they're called chloroplasts. I want you to think of chloroplasts as tiny factories that a plant uses to produce, aka convert energy into a usable form. And so it takes energy from the sun and converts it into a chemical energy that is actually usable by plants. Now, we need to understand the anatomy of a chloroplast. If we don't understand the anatomy of a chloroplast, all of this other parts of the conversation is going to be very difficult to understand. The first thing you need to know is that it has two membranes. And you should remember that it has two membranes because it was likely a part of an endosymbiotic event. And so what do I mean by that? I mean that it is very likely that chloroplasts were once free-living prokaryotes that were ingested by a eukaryotic-like cell, and it got its outer membrane from that endosymbiotic event or that phagocytic event, which allowed it to develop this second membrane. And this is going to start making a whole lot of sense for why it's able to be so good at making energy. If it can make its own energy, that's very likely makes it possible to have its own or be its own free-living cell. Okay, now we talked about the hallmarks of a cell in class. It has to have a cell membrane, it needs to have DNA, it needs to have ribosomes, and it needs to have cytoplasm. Well, we know it has a membrane. In fact, it has two. We know that it has DNA. We know that it has a ribosome, or lots and lots of ribosomes. Now, the one thing we didn't talk about is the cytoplasm. And the cytoplasm for this is basically called the stroma. So the stroma is basically the cytoplasm of the chloroplast. Now, the chloroplasts have other parts to them called the thylakoids. So these thylakoids are basically a little disc. I call it a pancake. That's not very biological, but it's this disc thing that actually had its own membrane where those light reactions occur. Now, if I stack these pancakes on top of each other, we call it a granum. So a granum is just a stack of thylakoids. Now, because these thylakoids have their own membrane, they have their own liquid inside of them, and that liquid is called the lumen. So, just so we're super clear, the chloroplast is part of a large plant cell, and a plant cell could have upwards of 40 chloroplasts in it. The chloroplasts are double membraned because they came from endosymbiosis. And inside the chloroplast, there are these discs called thylakoids, which are membrane bound themselves. And so these thylakoids have their own membrane that have their own liquid inside of them called lumen. And if I stack a bunch of these thylakoids together, that's when I have a granum. The thylakoid membrane is the site of the light reactions, which we will talk about in another video. So I think this picture is really cool. This is just an actual image of a chloroplast. You can actually see some of the features of the chloroplast. So we have the thylakoids here. And so this whole thing would make up a granum. And yes, it's a very crowded organelle. It's not just a bunch of empty space around like a single stack of thylakoids. There's all these other proteins involved, which we don't really get into in this course, and I actually don't know much about. Okay, so I told you that the thylakoid membrane is the site of the light reactions. And the light reactions can only occur because of something called photosystems. Now, photosystems are going to be a group of pigments and electron acceptor molecules within the thylakoid membrane. And so this image over here is really one of my favorites. It is going to show you the photosystem generally. And so you can see all of these pigment molecules. In this case, we're going to be talking about chlorophyll. And so it houses a bunch of these pigment molecules called chlorophyll in something called the light harvesting complex. The light harvesting complex is going to be able to pass energy from that photon that hits one of these chlorophyll molecules from a bunch of different chlorophyll molecules to the special pair. And this special pair is going to be very important. Okay, now there are two types of photosystems, photosystem 1 and photosystem 2, but I really would rather you memorize them in the other order because the light has to hit photosystem 2 first. So remember, when I said it in class, that photosystem 2 was discovered second, but it actually is hit by light first. So you need to understand when we say photosystem 2, it's really the first one in the electron transport chain, and then light has to hit photosystem 1 to re-energize that electron, which we will see in the next video. Okay, now let's talk about these pigments. These pigments are going to be something that absorb certain wavelengths of light. So there's lots of pigments. You don't need to know all of the pigments because there's way too many for you to ever know. But I do need you to know chlorophyll. So I want you to think of chlorophyll as basically a solar panel. It is going to be the thing that is able to absorb the light and then pass the energy around to other chlorophyll molecules. Chlorophyll is going to be found in the thylakoid membrane at the photosystems. Now, there are other pigments that I want you to be familiar with, but maybe not have to answer really a whole lot of questions with them. But carotenoids are going to be also photosynthetic pigments. Anthocyanins are not photosynthetic, but they are protective pigments. And the main reason I want you to sort of be familiar with these is because, for example, if you saw an FRQ that talks about carotenoids rather than chlorophyll, I don't want you to be panicked and say, well, we never learned it. And you're right, we never went into detail with it, but the ideas are the same, and that it is still a photosynthetic pigment, and it can be treated somewhat similar to chlorophyll. Okay, and so we have these absorption spectrums that are related to the pigments. And so chlorophyll, you can see it absorbs everything but green. So I could, for example, ask you, based off of an absorption spectrum, what lights are being reflected, or what wavelengths of light are being reflected, and what is being absorbed. And you should be able to tell me how that would appear to the human eye. And so chlorophyll, it absorbs everything but green, so it reflects green, and that's why we see green. Now carotenoids, they reflect greens, yellows, and reds, and so those typically appear orange to our eyes, and that's why we say things like carrots are very heavy in carotenoids. Okay, now chlorophyll is a really interesting molecule. I don't need you to know the chemistry too much about it, but I do want you to notice a couple of things. One, the hydrocarbon tail of chlorophyll. Now chlorophyll has this hydrocarbon tail, and it has a different head group, which you may remember is kind of similar to a phospholipid. Now this head group is not particularly polar like we've seen in the phosphate group, but it does get embedded into the cell, or not the cell membrane, the thylakoid membrane, to form that light harvesting complex. Okay, the last thing we're going to look at here is just these pigments. So we have the chlorophylls, which are going to appear green to our eye. We have carotenoids, which are going to appear yellow. And then we have anthocyanins that are kind of like our melanin. They're not particularly similar in terms of compounds, but I do want you to understand that they are both going to be protective pigments. That's all we have for this video. It's a very short one. The next one is going to be a little bit longer because it's a little bit more intense, but I hope this gives you a decent overview on the machinery needed, where things are going to be happening, and a good setup for how we're going to approach the light-dependent and light-independent reactions. Good luck!